Good morning. Today is Tuesday, May 26, 2020. My name is Ricardo Sandoval, and this is the results video for Laboratory 2, Aseptic Technique and Culturing Microbes. This is for Biology 225, Microbiology over at Los Angeles Pacific University for the summer term 1. I'm in my office today versus the garage, since this is my incubation location, and it's where the 3 petri dishes for E. coli, S. epidermis, and S. cerevisiae have been sitting for the past 3 days, as they did require an extra day to incubate, develop properly, so that I can actually see results on the nutrient agar that I poured in the past couple of days. And here's just a bit of quick little sign here, just summarizing what I just said as far as the lab data, all that good stuff. So, as we recall, 3 days ago or so, what I did is I had previously cultured in a nutrient broth, E. coli, S. epidermis, and then the S. cerevisiae. The S. cerevisiae came from yeast, and that was brewed in some warm water, while the S. epidermis and the E. coli rather came from developed capsules. So I'm going to show you basically here in a minute the results of the 3-day incubation. So just one quick thing I want to talk about here are the shapes that bacteria, microbiota, normal flora grow in. And there are 2 main types that we're going to see here on each of these 3 petri dishes, and that's what's called bacillus singular, or bacilli, and that's bacilli is plural, and that's a rod-shaped bacteria. And then there's also coccus, or cocci. That's a circular form or a circular shape of bacteria. So the first petri dish I'm going to show here is my E. coli, and as we can see, I have growth throughout the entire dish, which I'm very happy to see. Again, this is my quadrant 1, 2 is over here, 3, and then 4 is right here. So, I mean, as expected, the heaviest growth is in the top, or the first quadrant, because that's where, after I had dipped my inoculation lip into the broth, that's the first one, and then subsequent ones weren't as heavily populated, if you will, with the E. coli. And then, as we can see here, as far as shapes, you can see a lot of it's, well, down here it's the cocci shape, and then we have a lot of the bacillus, which is kind of hard to see because it's very continuous. But in doing some brief research prior to recording this lab video, I mean, this is pretty much what E. coli should look like once it develops on nutrient agar. Okay, so the next dish I'm going to show here is my S. epidermis. And then, as you can see here, pretty much growth here completely is the circular, or cocci shape here. And again, the most heavily colonized is the top of the dish, or quadrant 1, kind of diminishing for quadrants 2, 3, and 4. So that's S. epidermis, and again, S. epidermis lives on our skin, and really only becomes a problem if we have open wounds where catheters are inserted, if we're in the hospital, for example, for an IV catheter. And E. coli is also normal flora, normally found in our digestive system, which can also be a problem. It can become pathogenic with tainted foods, things of that sort. Okay, so the last dish here, this is the S. cerevisiae, and again, this is very heavily colonized. And this is the sample that came from yeast. So, I mean, you can see some of the round or cockeye shaped here at the bottom of the dish, down here. And then there are also some bacilli or rod-shaped growth here. But from my angle, from the bottom of the dish, I'll kind of flip it, see if that kind of makes you here easier to see. There's a lot of cockeye, but given, I guess, the density up here, I mean, they're kind of hard to see. But I'm very happy to see that all three cultures did grow as I expected. They spent 3 days and a couple of hours in a dark, temperature-controlled closet in my home office. I did not experience any difficulties with performing this laboratory, which I'm happy to say, given that this is my first my first attempt or my first experience with pouring agar and culturing microbes in this way. But again, this was my results video for this lab, aseptic technique and culturing microbes. Thank you.